Hello there. This is Dr. Dan Guerra coming to you from the beautiful Northwest from Vera Med Studios. Today, the 27th of October, 2017, I'd like to spend some time talking to you about appetite control. So the title of my talk is going to be The Road to Appetite Control. And as you can see from the initial slide, I'm showing you a nice mountain highway. This one happens to be up Logan Canyon near Logan, Utah, where I did my doctorate degree some years ago. At any rate, what I want to try to do is carry you through some of the newly discovered endocrinology and metabolism that associates diet with, first of all, appetite control, and that would mean the axis of association between the digestive system and the central nervous system, that is the brain, and also how fatty acid sensing and fatty acid uptake in the gut regulates appetite control and perhaps also adipose deposition, which of course can lead to weight control. Okay, so that's the basic framework I want to get into. I know that some of those that listen don't have a background in metabolism or in biochemistry for sure. So what I try to do in these are talks really geared towards general audience, is try to get you to the point where you see some of the nomenclature we use and help you to understand what scientists do to discover how appetite, in this particular case, how something that we think we know something about, like appetite control, is regulated at the fine-tuning level, at the molecular level, okay? And once we learn that, then possibly we can find ways to control appetite. Probably the least one that you'd want to go to is pharmacological, but maybe there are other considerations that we can understand. And also the genetic background of the individual people and why some people gain weight on a given diet and some don't. And also why some people can lose weight on a given diet or exercise regimen and others cannot. So the basic research is necessary before we can get to those larger, lofty, grandiose understandings, right? So that's what I'm trying to do here. All right, so let's go to the first slide. And of course, I'm at Vera Med Studios, where science meets biomedicine in a consultation format. So, obesity is a health crisis. I think we all know this. Many avenues of research are drawn to drive down this epidemic. That is research at the molecular level, the kind of thing I'm going to talk about to you this afternoon. Drugs targeting comorbidities are a major focus. What do I mean by that? That means that we have pharmacological interventions that relate to what happens after people become obese. What's a major player there in terms of a disease? Type 2 diabetes. But there are many others, right? Cardiovascular disease and cancer are two other major ones that are leading causes of death, of course. Surgery is an option for gross obesity, but may have side effects. Now, that's going to be a separate issue I'm going to bracket off today and not talk about. But I'm going to talk to you about jejunal bypass type surgery, gastric bypass surgery, and give you some details about what the current research says about it, the pros and cons. Okay, but we're not going to talk about that today. I'm just mentioning it. Prevention is probably the most neglected approach in the research community. That's because when you're conducting research, often you're using animal models, and you're not necessarily looking for appetite control, although this has been studied quite extensively in mice and in rats, and also indeed in human beings. So perhaps, though, we need to go back and look at prevention itself as a means by which to help us control this epidemic. I'm not going to talk much at all about prevention today, because some of that is really psychosocial, right? Today, I'm really going to talk about what goes on, again, between digestion and the brain, right? So that's really the key thing here. All right. Now, what do we know about feeding and hunger control at the neurological, endocrine, and metabolic levels? Well, we know a great deal. Obviously, in this short talk today, I'm not going to be able to give you what would be at least a one-semester course. I'm going to try to really redact it and just talk about a few things that are relevant to today's discussion. One of the things that many people believe is that lipids, that is fats in the diet, cause obesity. Now, is that true? Well, before I would even answer that question, I have to examine the contents of that query. And when I do, I discover, as a biochemist, that the question itself is poorly framed. You can't just ask a question like, does this particular food type, that is fats in the diet, one of the three main nutrients, carbohydrates, proteins, and lipids, are natively called fats. You can't just ask that question without asking a lot of corollaries to that question. And those correlatives would then relate to what kinds of fats you're talking about, what else is being consumed when you consumed fats, what's the exercise regimen, what's the genetics of the patient, and whatnot. So it's the same thing, of course, can be asked about carbohydrates and proteins. I'm just saying that if you pose a question like, do lipids cause obesity, it's a loaded question. But let me try to answer some basics about it, okay? First of all, excess caloric intake contributes to obesity. So anything that involves more calories than necessary for basal metabolism and normal just replacement of tissues like muscle is going to result in an increase in weight. And because of the fact that we are oleaginous organisms, that is, we put on weight in the form of lipid, that is adipose fat tissue, right? Depot fat, we call it. Obviously, anything that's more calorie in the diet than necessary for normal basal metabolism is going to increase obesity. And obesity then largely is adiposity. Adiposity means more adipose. Okay? And that's because of the nature of the human being. We are oleaginous organisms, which means we store fat. Okay? So decreased energy expenditure contributes to obesity as well, right? So that's intake, and that's also what? Utilization. So if you lead a sedentary lifestyle, you're going to gain more weight than if you have a more active lifestyle. So these are things that we've known, and I'm sure all of you know quite well. Now, excess protein and carbohydrate and lipid will contribute to obesity. And they will also disrupt metabolic homeostasis. Excuse me. So what do I mean by the latter part of that sentence there, or statement? Well, not only do any of the three major food groups contribute to obesity, and indeed adiposity, because protein and carbohydrate are converted to fat in the body when they're in excess, but also the intake of those food groups will disrupt metabolic homeostasis. And by that, I mean your normal metabolic regimen that allows you to carry out protein synthesis and gene expression and DNA turnover, and also, for example, the entire immune system and all of your senses and the control of those senses and the energy required to do muscle movement, contraction, etc. All of that can be disrupted by what you consume, how much of it you consume, when you consume it, and then all the other kind of things that are related to it, such as what's your gender, what's your age, even sometimes associated with ethnicity. Right? Again, these are general things I think that we know, but I want to make sure to bring it to fore that we know this also at the molecular level. So dietary lipid actually, amongst those three food groups, is more prominent in controlling obesogenic responses to overeating and sedentary lifestyle. That is, fatty acids are the sensor for whether or not a body is going to gain weight in a specific way and whether or not a body will lose weight relative to the amount of adipose tissue. Now, that's just because fatty acids are the component of the triacylglycerol that's in the adipose. That's of course something very significant. But the important thing here is that fatty acids act as sensing molecules or signaling molecules in the diet, as we'll see in my talk today. And so the consumption of fats in the diet not only contribute to the calories you consume and the energy you're able to garner from those calories, and also whatever gain of weight you might have or not based on exercise and caloric intake, but also they're going to control at the hormonal, that is the endocrine level, and the metabolic level, whether or not these contributory factors of diet and exercise are going to lead to more adipose, more obesity, and then all of the correlate diseases related to them. Okay, so what we're trying to say here. Now, diabetes is actually a fallout from obesity. That is, it's a disease that's associated with it. That's also, we can call it a fallout. It's now, diabetes now, because largely of this obesity epidemic, is also becoming a worldwide epidemic. Okay, diabetes may be affecting as many as 350 million people. That's a considerable amount of people compared to the 7 billion there are, right? It's quite a percentage. Remembering that like at least 1 billion people are starving in this world. So really only 6 billion people are the ones that are reasonably well fed. So 350 million is a lot. And that's how many people probably, in a conservative estimate, have type 2 diabetes right now. It's linked to obesity in many ways. Now, since hyperglycemia, that means high levels of glucose in the blood, is a primary cause of diabetic complications and accounts for the majority of the economic costs associated with diabetes, therapeutic approaches, that is particularly pharmacotherapeutic approaches, are focused on regulating glucose levels and on something maybe now you're starting to learn something that probably you don't know from the common literature like newspapers and magazines, or certainly from the TV or the internet, hepatic glucose production, or GP, glucose production. So not only do you have to be concerned about circulating glucose and how that's related to diet, but you have to think about the fact that your body synthesizes glucose, particularly your liver. And that process is called gluconeogenesis. Okay, that's the pathway leading to the synthesis of glucose from non-carbohydrate precursors or substrates. Okay? That's what gluconeogenesis is in biochemistry. So when you think about the drugs that people get when they come into a clinician, into a medical clinic, and they talk to a clinician or a nurse practitioner, and they're obese and they're complaining about various morbidities that may be associated with common diseases like diabetes. If they don't pass sufficiently an oral glucose test, and it's determined that they have diabetes or that they're pre-diabetic moving to diabetes, a drug that's often administered, right, a pharmaceutical, is called metformin. So that is probably the most widely prescribed initial diabetic drug, right? And what that drug actually does is it reduces glucose production. Now, in previous talks, I've discussed this, okay, how that works. It has to do with the fact that metformin attacks the complex 1 in the electron transport chain, which involves the oxidation of NADH to NAD, transfer of electrons and protons through the intermitochondrial membrane and out into the intermembranous space between the outer and inner membrane of the mitochondrion, thus ultimately leading to ATP synthesis. I know that's a lot to say, and if you're not a biochemist, which probably most of you aren't, even if you are educated at some level in metabolism, that might be a mouthful. Basically, what I'm saying is that standard normal control of energy production, okay, what you do when you take metformin is you're helping block energy production because it takes energy in the form of ATP, adenosine triphosphate, to make glucose. Okay, so if you block energy production, you don't have the energy necessary to make glucose, because to synthesize glucose, to make it in the body, you need energy, metabolic energy in the form of ATP. That's how metformin works. And again, previous talks, I've gone into much more detail than that. That's all you need to know now. Now, metformin has side effects, as you might guess, because it's blocking energy production, right? It's not just affecting glucose levels, right? That's your layman's understanding of it. You don't want to be a layman anymore. You want to have more information. You want to gain knowledge, right? Of course you do. So metformin has side effects, And that leads to the development of hypoglycemic agents aimed at increasing circulating levels and the action of gut-derived hormones called incretins. Okay, so the fact that taking metformin can actually lead to hypoglycemia. Okay, so you can actually get lower levels of glucose than is necessary. You pass the mark and you start having lower levels of glucose. You have to then look at all of the possible downstream effects. So there's an enzyme called dipeptidyl peptidase 4, DPP4, and one of the drugs that's also administered in terms of controlling blood glucose levels after going on an initial regimen of metformin is an inhibitor of that enzyme. And also glucagon-like peptide 1 receptor agonists are another class of compounds that are used. So if you're a diabetic and you've already gone on metformin and you're still having a problem with glucose control, maybe you're just having episodes of hypoglycemia, to help control that, you move further down the stream, the pathway of what metformin is affecting, okay? Or maybe not directly affecting, but correlated to that effect of what metformin is doing. You now have to go downstream from those pathways and control the effect of the drug, okay? And so you have new drugs then that relate to that. And so this is all going to get us heavily into endocrinology, right? So there's a couple of the pharmaceuticals that are actually being prescribed when there's associated effects of initial pharmacotherapy associated with metformin. Okay, and there they are. So the early success of all of these treatments highlights the importance of the GI tract in regulating glucose homeostasis. Now, if you remember, if you play back the tape right now, I said that gluconeogenesis, synthesis of glucose from non-carbohydrate precursors, primarily is a hepatic function. Hepatic is a fancy word for liver. That means that the liver is the organ that does this primarily. So what are we talking about here? We're saying the GI tract is involved. Well, the GI tract is not just the liver, right? The liver is associated with that. But here we're talking really about the small intestine, okay? So we have to understand that in the digestive process, many organs are controlling what ultimately relates to nutrient homeostasis. And one of those nutrients is glucose. And that's associated with diabetes, which is associated with obesity, you see. So what about the GI tract? It's anatomically positioned to prevent nutrient excess via a negative feedback mechanism through nutrient-induced secretion of hormones. So that is, as the GI tract is receiving, right, incoming nutrient, that incoming nutrient sends a hormonal signal, an endocrine signal, to stop feeding, okay? So part of that has to do with gastric emptying, the stomach emptying its contents, hormones that regulate that, because if you slow gastric emptying via gut-derived signaling, what's that going to do? It's going to make a person feel full, right? So if you feel full, what normally happens? You stop eating, right? So you see how this is related to just when you sit down and have a meal, right? You have your steak and potatoes and you have your carrots and you're sitting there and you're feeling pretty darn full, right? You just had 12 ounces of distilled water or some other kind of beverage with no calories. You feel pretty sated, you feel pretty full. But then somebody comes along and says, would you like a piece of apple pie? A lot of people say, well, give me a little while to digest this, right? Well, that digestion process means that you feel really full after that big steak dinner, right? Well, that feeling full is supposed to stop you from eating. Now, we all know the problem is that even when people are full, and even when they're not trying to be polite to somebody that's offering them pie because they're at someone's party or something, even when they're at home, they may still have ice cream or pie or whatever else, right? After dinner or shortly after dinner when they really don't need to have any more nutrients. So there's obviously something else going on besides simply that the GI tract stops you from eating. So what that something else is related to all this discussion we're going to get into right now. So in the stomach, there's a hormone called ghrelin, and it's what we call an orexigenic peptide. And what orexigenic means is it stimulates the appetite. So when ghrelin is made, it makes you hungry. Okay, it makes you hungry, right? And that's synthesized primarily in the stomach. So we're going to talk a little bit about ghrelin today. I don't like that. I know that word is probably foreign to many of you, but it's a very important hormone. It's not the most important hormone in controlling appetite. There are a lot of other ones involved, as we're going to see, but it's one of the major ones, and it's gotten a lot of publicity in the last, almost decade. Now, what does ghrelin do? Ghrelin increases are associated with the timing of a meal, while nutrient ingestion suppresses ghrelin secretion. So when you take in food, say you went to lunch and you had a, barbecue beef sandwich and French fries, right? What's going to happen to ghrelin synthesis and secretion is it's going to tank. It's going to stop. It's going to be suppressed. Okay, so right away, I want you to see this cause and effect. That's just at the stomach level. Now, although ghrelin may act in an endocrine fashion, that means it gets into the bloodstream, and once it gets in the blood, it binds to certain receptors peripherally in the central nervous system and muscle tissue, or in the gut and various other regions of the body. And in so doing, it helps regulate overall nutrient homeostasis. That's an endocrine type of effect. It means it's in the blood and circulating in multiple passes in circulation. What's been discovered is a vagotomy, which disrupts the direct communication between the stomach and the brain via the vagus nerve. A vagotomy does that. It actually abolishes much of the ability of ghrelin to increase food intake, suggesting a paracrine effect. Now, paracrine rather than endocrine means that the association of the synthesis of the active compound, in this case ghrelin, and the utilization of it does not require blood circulation. Okay, so paracrine means it's from a cell type or tissue or organ to one other cell type or tissue or organ, and not general circulation. That's what a paracrine effect is. And we know that because if you take care of the vagus nerve, you block it. So that means right off that ghrelin is affecting something directly in the brain, because that's where the vagus nerve goes. Okay? So ghrelin also regulates glucose homeostasis by increasing the gastric emptying rate and inhibiting glucose-stimulated insulin secretion. Okay, so ghrelin has a lot of effects. Now, there's more of the endocrine effect. Okay, now actually, it's not ghrelin; it's what we call acylghrelin. This is the actual potent hormone. So it's actually post-translationally. That means after you make the protein. First, you transcribe a protein from DNA, you make messenger RNA, and the messenger RNA is translated to protein, okay? Once you make that protein, and ghrelin is a peptide hormone, so it's a protein, you have to covalently modify that protein by adding an octanoic acid. An octanoic is an 8-carbon saturated fatty acid. You have to add that covalently to a serine hydroxyl group on the ghrelin molecule. So we call that post-translational octanylation. Okay? Once you add that octanoic acid, now you have the active acylghrelin that is the most active. Actually, desacylghrelin, that is without the octanoic acid as serine 3, may have opposing and unique endocrine effects, especially in the brain. So adding or subtracting that fatty acid to that serine residue on that peptide regulates ghrelin activity, maybe even at the level of what valence, that is up or down, right, or on or off. See, that's what I mean by valence. Okay. So further proteolytic degradation of the ghrelin to smaller peptide fragments can occur in the blood, actually, and that removes the mature 28 amino acid hormone from biological activity. Okay, so I'm giving you this detail so you get an idea about how much we actually know about something that relates to gastric emptying, as well as whether or not you have an orexigenic stimulus in the brain. And that is directly related to this acylghrelin metabolic activity, right? So I'm just covering that off now. That's not a main emphasis of today. I'm just giving you this clear indication about, we know a lot about acylghrelin, and we know about how it's turned over, and we also know how it modifies appetite. Okay. So let's go back to talking about the GI tract. Small intestine contains a variety of regulatory signals. One of them is CCK, which I will detail in a moment. And that's found in so-called I cells. There's another hormone called glucose-dependent insulinotropic polypeptide, or GIP. That's in another type of intestinal cells called K cells. And in the ileum and large intestine, moving away from the small intestine now, and what are called the L cells, we have a third peptide hormone to consider, glucagon-like peptide 1/2, or GLP-1/2. We also have oxyntomodulin, or OXN, and peptide YY, or PYY. So there's 5 different regulatory signals, all coming from not the stomach now, that was the ghrelin story, but directly from the small intestine and from specific cell types, okay? So you get to see the complexity here. That's what I want you to see. There's a great deal of complexity here. And this complexity is in association with appetite control. Ultimately, it can also be overrun, and you get obesity. Okay? That's what we're talking about. Remember that, keep your eye on that major consideration. We're talking about obesity and appetite control. Okay. Now, the secretion of these hormones is stimulated by nutrient ingestion, which you might guess, right? Within the intestine, that's where this is happening. Then they act on their respective receptors. So you have these proteins binding to another protein called a receptor on the surface of cells that are responsive, right? And those receptors either centrally or locally or even vagal afferents. Afferents just those are the cells associated with the binding of the stimulation of these hormones that come from the GI. Okay. Now, all of that's in close proximity to those enteroendocrine cells, right, where these hormones are synthesized. And this is all associated to regulate the metabolic homeostasis through various changes in food intake, as the appetite control, gastric emptying, which of course indirectly controls whether or not you feel hungry, and intestinal motility, which has to do with the absorption of the nutrients in the gut, okay, the motility we call it, and or energy expenditure at some other level, right? Like how much energy is expended through the digestive process, but also how that nutrient storage is ultimately used in terms of the body's activity. So you see here in this slide, this pictorial here. Here is the upper intestine, the small intestine. There's a duodenum. And here are the hormones the duodenum is involved in governing the synthesis and secretion of: CCK, GIP, GLP, and OXN and PYY. Now, there's some question marks here because we don't know exactly about how it controls secretion, but we know it's associated with it. And remember, the stomach is proximal to or above the small intestine, right? Now, another part of the small intestine directly below or distal to the duodenum is the jejunum. And in the jejunum part of the small intestine of the GI, CCK and GIP are also synthesized, secreted. And then all the way down the last portion of the small intestine of the GI is the ileum. So you have the duodenum, the jejunum, and the ileum. A little bit of anatomy here. The GLP-1 and 2, OXN and PYY are secreted. So you can see here that there's metabolic zonation or localization of where these hormones are secreted. And it moves down the digestive process. So as you digest food and you're absorbing some of that food in these various tissues, right, in these various absorptive cells in the small intestine, as the nutrient passes through and contractility increases, right, absorption increases, you're altering the menu of hormones that are being synthesized and secreted into the blood along with the absorbed nutrients, sending signals back to the brain, controlling things like appetite. See? So your digestive process is signaling to the rest of the body that what's coming through, what kind of nutrients are coming through, protein, carbohydrate, lipid, how much of them, and also what is the discrete quality of those three macronutrients. That is, what kind of fatty acids, what kind of carbohydrates, what kind of proteins, right? So that's all being sensed in your GI. And all these hormones that are going to be expressed and synthesized and secreted according to what you've consumed, right? And remember, the stomach is involved in ghrelin, also another hormone, leptin, which is an adipokine. And we won't talk about it, that today. I've talked about it previously, and we'll talk about it again some other time. Now I want to talk about these hormones that probably you never heard of, right? And if you have, of course, if you've been educated, if you're a medical doctor, or if you're a scientist, or you're in graduate school or something like that, you have heard of these. But even if you have, let's refresh. And many people that are watching this have never heard of these. But I want you to know these are big players. These are vital components of your digestive system and appetite control. And when you think about pharmacotherapy, when you go to a clinic and a physician gives you drugs, some of the drugs that are coming on the market from the pharmaceutical companies are going to regulate these hormones, okay? If you learn more about these, you'll learn more about mode of action of the drugs you may be prescribed. That's why we're doing this at Vera of Med, you see? Okay, so let's talk about glucose-dependent insulinotropic polypeptide, G1P. G1P is a 42 amino acid, so it's not very long. It's a peptide. It's a hormone that is produced by the enteroendocrine K cells, a specific type of cells in the small intestine, released in circulation in response to nutrient stimulation. Okay, we already know that. Both GIP and GLP, and I'm sorry that they look very similar, okay, but these are the redacted acronyms for these two hormones, these gut hormones, GIP, GLP. They both serve to stimulate insulin secretion in a glucose-dependent manner and are thus classified as a subclass of hormones called incretin. They increase insulin. See, increase insulin. That's why they're called incretins. Pretty fancy, right? These endocrinologists, I think, are really good at naming things. I won't say that biochemists are so good at this. In fact, we're pretty bad at it. But we're a heck of a lot better than the geneticists who make up really goofy names for things. But we'll leave that for another discussion. All right. So anyways, those are called incretins, okay? And you see what they're doing. In a glucose-dependent manner, they're stimulating insulin. And remember, insulin is yet another hormone synthesized in the beta cell of the organ called the pancreas, right? And what it's doing, I mean, it does a lot of things, okay? And saying that I know sounds way too generic, but insulin is a very, very significant circulating hormone. Controls things like protein synthesis and turnover of nucleic acids. It's a very potent and highly facilitating hormone. But for right here, we know that insulin secreted from the pancreas is going to do what? It's going to cause glucose uptake from the blood. And why is that significant to our discussion today? Because when you take up glucose from the blood, which is supposed to happen after you have food, that's going to remove it from circulation, and it's going to make it so that you utilize that glucose. Remember, high levels of circulating glucose that don't go away after a meal, hyperglycemia, that's the hallmark of diabetes, right? High circulating glucose. Hyperglycemia is diabetes. That's the major biochemical phenotype of it, right? Now, these hormones here are going to cause GIP and GLP coming from the gut are going to bind to the pancreas, and they're going to cause insulin to be secreted as long as there's glucose available. That's what it means by glucose dependent. High level, you have to have glucose as well as these hormones, and they, and also in league with fatty acids, are going to cause insulin to be secreted, which is going to pull glucose out of the blood, put glucose into those tissues like muscle and adipose tissue, lower serum glucose levels, and keep you from being diabetic. So following secretion, GIP is metabolically degraded by that protein we already described to you. This is an enzyme. dipeptidyl peptidase 4, DPP4. And we told you that there are drugs associated with that. Why? Because if you inhibit, look at here, if you inhibit that drug, remember I told you antagonists are being administered as pharmaceuticals. If you inhibit that enzyme, right, DPP4, you're going to prevent the degradation of GIP, and that's going to allow GIP to continue to stimulate insulin secretion, which is going to lower what? Glucose levels. See how that works? Okay. Now, in addition to its insulinotropic, that means it causes insulin to be turned on, put into the blood, GIP exerts a number of other actions, including promotion of growth, survival of the pancreatic beta cells themselves, and stimulation of adipogenesis. So GIP also stimulates the synthesis of new adipose cells, adipocytes, in the depot fat. Now, why are those linked? Because if you're getting nutrient, I told you we're oleaginous organisms, ultimately all that nutrient, big time carbohydrate like glucose, is going to be taken up by the adipose, okay, and it's going to be converted to fat. Okay, so that's the whole point. You're going to convert carbohydrate, excess protein, and then the fatty acids themselves that are in the diet are all going to be ultimately stored in the adipose. Okay, and this hormone from the small intestine is going to help regulate adipogenesis, or that is the synthesis of new adipose. So it's really well linked and really well described. GIP also increases the expression of something called anti-apoptotic BCL2, and that decreases the expression of the pro-apoptotic BAX protein. That results in a reduction in pancreatic beta cell death. So anti-apoptosis, apoptosis means programmed cell death. So if you increase the expression of an anti-apoptotic protein, at the same time decrease the expression of a pro-apoptotic protein, you're going to get less beta cell death. And that's specifically where GIP is functioning. Because remember, there's a receptor for it there. Okay? So it's going to maintain beta cell life, beta cell integrity. Pancreatic beta cell is what gets degraded either in an autoimmune response in type 1 diabetes, or eventually because of chronic hyperinsulinemia, type 2 diabetes. Okay, so if you can maintain beta cell vitality, that's a good thing. And that's what GIP does. You see, so it's a very important hormone coming from the gut, sensing nutrient. See? Now, in adipose tissue, that's pancreas, GIP interacts with insulin to increase lipoprotein lipase. That's taking fatty acids out of the blood, those things called lipoproteins. Lipoprotein lipase is an enzyme that removes fatty acids from circulating lipoproteins, right? And in so doing, because it's adding those fatty acids out of the adipose, it induces lipogenesis. Lipogenesis means the synthesis of lipid. What lipid? Triacylglycerol, right? Major storage form of fat. TAG, we call it. Triacylglycerol, not triglyceride. Anybody that says triglyceride should be put in the corner and told that they have to put a dunce cap on for at least 10 minutes. That seems kind of harsh, but I went to Catholic school, so believe me, that's not as harsh as it got. Okay, but the reason I'm saying that is you don't say triglyceride, because triglyceride sounds like there's three glycerol molecules. No, it's only one glycerol molecule, and there's three fatty acids. So we say triacyl. That's the radical form of the fatty acid that is in an oxygen ester to the glycerol backbone. Glycerol, triacylglycerol. So call it triacylglycerol, not triglyceride, please. Because you don't want to do that dunce cap thing. So there we go with that. Now, there is a significant interest in the potential clinical applications for GIP analogs, and both agonists and antagonists have been developed in preclinical studies. Now, why would you want to antagonize it? Because maybe you don't want too much insulin secretion, right? So remember, it's a set point. Hormones are all working, all of metabolism is about homeostasis. So it means there's a set point. And you want to get a set point in the body. You've probably heard this if you're a person that's in, if you go to the gym, right, and you work out, the trainers will say, well, you need to get to a set point where you have a certain basal metabolic rate, you have a certain VOX, right? The amount of oxygen consumption when you're working on a treadmill. You want to get to a set point, heart rate, right? Respiration, all that, right? For exercise physiologists and trainers, we know all about this, but also the people that go to the gyms. Well, that set point's the same thing that happens metabolically and happens at the endocrine level. So you want to have a set point, and that means that you don't always want to stimulate a hormone. You also sometimes want to antagonize it because some people go over the mark. And whenever you go over the mark with a hormone, you start going in the other direction. And whatever the hormone normally was doing, not only does it stop functioning, but because of feedback control, you get negative feedback. You might stimulate what used to be the consequent. So you deny the consequent, right? Which I know that some of you are very interested in when we talk about categorical logical statements, right? All right, so that was GIP. Let's talk about GLP. I know it's taken a long time, but this is really important. And what else do you have to do on a Friday afternoon, which is when I'm recording this? And of course, you're going to watch this right after it gets uploaded. I know. All right, glucagon-like peptide 1. It's synthesized in the intestinal endocrine cells. Okay, and there are two major forms of them, GLP-1 and GLP-2, and those are two different peptide fragments differing only by one amino acid. Okay, isn't that amazing? Now, GLP-1 secretion by the ileal L cells, right, the ileum, is dependent on the presence of nutrients in the lumen of the small intestine. Okay, so you need to get nutrients into the small intestine, absorbed, right, via the motility activation in the small intestine. GLP-1 increases insulin secretion, so it's an incretin. We already brought that to the fore. And it inhibits glucagon release. Now, glucagon is the other pancreatic hormone, and glucagon basically stimulates gluconeogenesis. So it's just the opposite of what insulin does, right? So when you're in a starvation state and circulating glucose drops, the pancreatic hormone that's secreted is called glucagon. And glucagon stimulates gluconeogenesis, okay, making more glucose, right? So you see how that works. Again, set points. Right? But GLP actually inhibits glucagon release. Now, GIP doesn't seem to have that activity, but only when glucose levels are elevated, thus offering the potential to lower blood plasma glucose while reducing the likelihood of hypoglycemia, okay? So it's going to inhibit glucagon release, all right? But it's only going to do that when glucose levels are elevated. Now, that's important. Why? Because if glucose levels were low and glucagon release was inhibited, you wouldn't make more glucose. You wouldn't be able to do that. You wouldn't be able to kick it back up. And what would you get? What would be the result? Hypoglycemia. Hypoglycemia is not a good thing either. It makes you faint. You could die, in fact, right? So bad player. So you see how these hormones, again, are always working bivalently, right? Bivalently means up and down, right? Or forward or backward, right? The valence. Gastric emptying is delayed and food intake is decreased after GLP-1 administration. Okay, so gastric emptying is delayed. That means your stomach holds the nutrient you just consumed longer. What does that do? It makes you feel full. Remember the whole ghrelin story too, right? It's going to inhibit ghrelin release. And it's also going to have an effect on food intake. Why? Because your stomach is full, so you're going to feel full, satiated. Now, in a 6-week study, investigating continuous GLP-1 infusion. This is now a human study. Patients with type 2 diabetes achieve significant weight loss. That's 2 kilograms. That's quite a bit because each kilogram is 2.2 pounds. So do the math. And a reduction in appetite from baseline compared with patients receiving placebo, where there was no significant change in weight or appetite. So if you do things like infuse GLP-1, right, directly into the blood, that's what infusion means here. Patients that already have full-blown T2D, type 2 diabetes, benefit from that because they get a reduction in appetite, which makes sense because of the gastric emptying thing we just talked about, for example. And also, it appears that they have an effect on weight, right? There's a decrease in weight. Look at that. That's like a weight loss of, Again, 2.2 times 2, that's over 4.5 pounds a weight loss for this 6-week study. I'm sure that most people that are on diets would like to lose 6 pounds in 6 weeks, right? Maybe it's not breathtaking, but it seems to be maintained, and it seems to be directly associated with this hormone. Yeah. So preclinical studies reveal other potential benefits of GLP-1. So receptor agonists, that means you're You're promoting GLP-1 activity. That's what it means to agonize something, not like when you're in agony, right? So when you agonize something, when you do hormone speak, that means that you push it forward, you push it along, right? You don't antagonize it, you agonize it. Same. All right. Sorry for those of you who know all this stuff, but many of my listeners don't. So it's a receptor agonist treatment in individuals with type 2 diabetes. Again, those are the patients, which includes promotion of beta cell proliferation, as we saw, and reduced beta cell program death, apoptosis. By the way, that's not apoptosis. Don't say that wrong. It makes you sound, again, the dunce cap thing in the corner. It's apoptosis, okay? That's a diphthong. The PT there is a diphthong in grammar. So you don't say that P. You say apo, which means before, tosis, which means what? Cleaving or dropping off, okay? In this case, that means what? Right? Apoptosis means right before you get what? Cell death. You see how that works, right? So that's actually a term. Tosis is actually a term from leaf drop in deciduous trees, like what's happening right now outside the window. All right. These include preclinical studies, as I just said, with people in type 2 diabetes. However, because of native GLP-1 is rapidly inactivated by that same enzyme that's circulating DPP-4, it has a very short half-life, a minute and a half. Wow, that really gets removed quickly. So that means it's a very potent hormone when you see that. To achieve clinical potential for native GLP-1, patients might require native 24-hour administration of GLP-1. That means you have to constantly infuse it. Or you do what? What we just talked about before. You inhibit DPP-4, right? So you maintain circulating levels of GLP-1 longer. But again, you have to be careful of that. You might overshoot the mark. You overshoot the mark, you're going to come into other issues, right? Not just feedback inhibition, but you don't want to overstimulate something. So drugs that are targeting these peptides, hormones from the gut, are very new on the market, and they're very potent, but we have to be careful about dosing. We also have to be careful about how long a person's on these drugs, right? So only in terms of looking at the obese type 2 diabetic and how long you can go on these drugs, lower their weight, get appetite control maybe, get them down to some set point, and then get these drugs out of there. Right? No drugs should be taken in maintenance like that. Certainly not ones that regulate directly these hormones. Third hormone. Sorry for so many, but there are that many, as you saw. CCK. That's cholecystokinin. Okay? Cholecystokinin. Love that word, but everybody wants to say CCK, so I'll go with that. That's a brain gut peptide. So in the gut, it induces the release of pancreatic enzymes. So once it gets to the pancreas not only is an endocrine gland, it's also an exocrine gland. Exocrine means it releases basically enzymes into the blood, enzymes and other things, right? Which we could talk about, like bile acids. Anyway, CCK induces the release of pancreatic enzymes, and it controls the contraction of the gallbladder, which is also important for digestion. In the brain, its physiological role is not quite clear yet, but it does have receptors in the brain. Okay, we'll talk a little bit maybe about that today. Here we go. Exogenous CCK exerts a variety of effects depending on the site of administration. Progress and understanding of the potential role in the brain and behavior has focused on 4 major areas. CCK seems to affect dopamine. Okay, now that's the reward pathway in the brain. Okay, makes you feel good, makes you feel mellow when you have a lot of dopamine secreted in the brain. So CCK has a control there. Doesn't that make sense that something coming from the gut is going to make you feel relaxed, right? When CCK is synthesized because of nutrient uptake, you feel good after a meal. You feel good. That's the reward pathway. It's a dopaminergic pathway. CCK then is also perhaps involved in anxiety and panic states, maybe less of it causing more anxiety and panic. CCK is also involved in opioid nociception. Nociception means pain reception. And as we've been saying, CCK is involved in satiety, whether or not you feel full. So it's very potent in the brain. And there's 4 main areas. So manipulating CCK, right, is going to have an effect on many aspects of brain-associated behavior, right, as you see there. Right? So you have to be careful about administering drugs that are going to hit this potent peptide hormone, this little CCK. So CCK prohormone, this is just a little lesson here. So you make a prohormone, you make a longer peptide, and it gets processed. And it gets processed by its own specific endo and exoproteolytic cleavages. That means it's cut down to smaller sizes. Endoproteolytic degradation means within the protein it's clipped. Exo means from the N-terminus or the carboxy terminus. Endo-exoproteolytic processing. Okay, or cleavage means cut. Two transcript variants encoding the same protein have been found for this gene. So you get two different transcript variants. So you have, when you have transcript variation, that means that this is regulated even at the transcriptional level. So again, not just the protein level, not just the protein turnover level, binding receptors, all that neat stuff you think about peptide hormones doing, right? That's how they function. But also it looks like there could be transcriptional variants, right? And when you have transcriptional variants, that means it's another level of control that we have to be looking out for, okay? CCK and gastrin, yet another hormone, induce the activation of something called pro-fibrogenic pathway, and that involves all these other enzymes. Now, these enzymes, I'm sure those of you in pharmacology, and certainly those of you who are physicians, and those of you who have anything to do with biomedicine know about AKT, ERK, and SERK. Those proteins sometimes are bad players in what? In things like cancer or even cardiovascular disease. So CCK has an effect on the activation or regulation of those proteins. Those are kinases, okay? And when those kinases get mutated, okay, there's a change in the amino acid composition because of a DNA mutation, they can become oncogenic, right? So again, pull back here for a minute. I want you to see manipulating CCK can have a lot of effects in the cell, not just in terms of nutrition and appetite control, obesity, etc. So you have to be very careful about modulating something like this. That's why I'm bringing it up. Okay. All right. AKT activation, for example, by CCK and gastrin could be inhibited by a particular kinase inhibitor called warp-mannan. So already people are studying this, right? Already people are studying how CCK and gastrin relates to controlling kinase activity. And when you control kinase activity in a negative way, that could mean elevating its activity, you could cause things like promote cell division, which could lead to neoplasia, lead to tumorigenesis. So there are actually already regulatory components out there controlling how CCK functions, right? Already in the pharmacopoeia. All right. Now, all these data suggest that CCK and gastrin are able to induce collagen synthesis in those cells, right? So therefore, they appear to be important regulators of pancreatic fibrogenesis. Now, what about that? Well, once you start making collagen and you start making or inducing fibrogenesis, okay, the next step there can be pancreatitis or inflammation of the pancreas. And you know what the next step is? Pancreatic ductal adenocarcinoma, right? Pancreatic cancer, bad players I've talked about in a previous Vera Med lecture. So this protein, CCK, appears to regulate in a way that we may not like when it's always turned on, this fibrogenesis in the pancreas. Remember I told you how it works, right? You get collagen synthesis, you get fibrinogenesis, that can lead to pancreatitis, inflammation of pancreas. And if all the signals go in the wrong direction, that can lead to adenocarcinoma, ductal carcinoma of the pancreas. And that can, of course, lead to death. So we have to be really careful about CCK activity. That's why I'm telling you now about this, because you can't just say, well, let's control this. It's going to help us with all this great stuff about obesity. Whoa, wait a minute, throw the anchor out. Throw the anchor out. We have to think about what else is going on if we regulate CCK. So CCK is secreted, back to this discussion, from the enteroendocrine or EEC cells, the eye cells, predominantly located in the proximal small intestine. Okay? And it's mainly in response to, look at this, first time we're bringing it up, fatty acids. So endogenous CCK mediates the ability of intestinal lipids to lower glucose production via a gut-brain-liver neuronal axis. Boy, is that a mouthful, right? Holy cow. So CCK, very potent hormone, right? But it's the one that likes to sense fatty acids in the gut, all right? So that's where we're going to be able to deliver now. We're about halfway through this talk. Sorry it's taking so long, but it's a complicated talk. But you'll learn so much if you play it back a couple of times. I hope. So this glucose lowering effect. Okay, it's going to lower glucose production of duodenal lipids. So fatty acids are going to knock down gluconeogenesis via CCK. It's dependent on intracellular esterification of long-chain fatty acids to long-chain fatty acyl-CoAs. Now, that's also a big mouthful, but in order for a fatty acid to be absorbed and to put in a metabolic play, to do all the things it's supposed to do, you have to make an ester of it. We already talked about the oxygen ester that you see when fatty triacylglycerol is made for adipose fat. Same kind of esters actually you find in many membrane phospholipids. When you make a thioester of a fatty acid, combining it with a coenzyme called coenzyme A, which is derived from the nutrient pantothenic acid, B vitamin, when you make that acyl-CoA, you put that fatty acid in metabolic play. And what this whole thing is saying here is that the fatty acids have to be esterified, make a thioester to coenzyme A via this enzyme called ACS. Okay, all of that is how you associate the glucose lowering effect of the duodenal lipid. So you need to have the esterification, which means that enzyme's got to what? Jack up to be able to put the fatty acid you just took in the diet to put in a metabolic play that ultimately, bing to bang, controls glucose production, lowering it. Okay. That indicates both the importance of both luminal lipid hydrolysis to long-chain fatty acids and subsequent absorption into intestinal cells, possibly via a transporter called cluster of differentiation. Whenever you see a CD in front of a protein, that comes from the old. Remember I told you how the geneticists are terrible? See, I like to trash or bash the geneticists because people always like to trash and bash the biochemists, probably because we're more clever than geneticists. And the physiologists like us a whole lot better. And physiologists are probably, well, if biochemists are like the top of the ladder of, science, and I don't just mean natural science, I mean all science, and that includes physicists and things like that, then right below us probably are the physiologists, right? So biochemists, then physiologists, and then geneticists, right? Okay. So anyway, cluster of differentiation, that's what that CD stands for whenever you see that in front of a protein. CD36. Again, don't worry about where that nomenclature comes from, Although the role of CD36, which is now going to be a protein, we're going to call it, it's going to be a protein on the plasma membrane, on a surface membrane, that's going to be associated with taking up fatty acids. So now we're getting into the fatty acid story. Okay. The role of CD36 in mediating glucoregulatory role of lipids, because that's what we just talked about here, it remains to be tested more carefully. It is interesting to note, okay, that if you knock out and you remove CD36 in mice, you exhibit a reduced release of CCK. So it looks like whenever you're affecting, okay, the CD36, which is involved in the uptake of fatty acids, which is involved in the turnover of the active fatty acids from acyl-CoAs, okay, because you need to get those acyl-CoAs hydrolyzed to just free fatty acid to get them to move through. All right. Whenever you disrupt that CD36, you disrupt CCK. See, so this is where rubber hits the road. This is where the fatty acids in the duodenum are more clearly related to what's going on with CCK synthesis and release. See? So furthermore, long-chain fatty acid-induced secretion and glucose-lowering effect of CCK are dependent upon a protein called protein kinase C delta, or PKC delta, stimulation, which is localized in the duodenal mucosal layer. Okay, so all of this stuff is really important because here it is. Long-chain fatty acids induce PKC delta, induce CCK pathway, and that requires the activation of CCK1 receptors, and all that's located on the vagal afferent fibers innervating the small intestine. This is how you get CCK poised to send out into that vagus system. Okay, got it. Binding of CCK to its receptors leads to PKA signaling, that's protein kinase A signaling, which enhances the NMDA receptor that mediates neuronal transmission in the nucleus of the solitary tract to lower glucose production via the hepatic vagal branch. So here we go. Finally, right? CCK, this is now way down into the weeds. Binds its receptor, turns on PKA, enhances NMDA, that's a neural receptor, and that mediates neural transmission. That goes to the nucleus of the solitary tract, and that ultimately lowers glucose production via back to the hepatic, because that's where you're going to get gluconeogenesis vagal branch. All these results suggest the paracrine, CCK is paracrine because it's going through the vagal tract, mediates the ability of the duodenal lipid to regulate glucose homeostasis by signaling to the brain, central nervous system. So there you go. See? Very complicated, not so very complicated, but more complicated than maybe what Occam's razor would want to allow. Okay, so let's put some of this together. All right. So here you got fatty acid signaling and hormonal appetite control. Okay. Here are your various intestinal cells. There's Mel cells. These are just plain old, and all these are enterocytes, right? This is where you're taking up nutrients in the small intestine. You got a lot of players here. You got free fatty acids. You got chylomicrons. You got all these hormones at play. Okay. Here are, this is a G protein coupled receptor of various molecular masses. There's GLP-1, there's GLP's receptor, right? You can see where these are localized when you look at where they are on these enterointestinal membranes, okay, enterocytes. What I want you to notice that all of this is mediated by that protein called CD36. So CD36, okay, that's what's mediating the uptake of these free fatty acids, okay? Mediating the uptake. That's all CD36 at play there, okay? That's the important thing there. Once you get those fatty acids taken up, see, there they are, right? That's where they're binding to the vagal afferent, right? And also they're binding to the this vagal afferent through this other system, the intestinal L cell, right? Ultimately then, because working through all of these receptors, GPR, GLP, CCK, all those hormones are at work here. What does that ultimately induce? Slowing of gastric emptying, so your stomach stays full longer, and in the brain, suppression of food intake. So you see how fatty acids are linked midway through all of these, the CCK hormones, as well as through GLP, okay, and through the G protein coupled receptors, which are required for the signaling to be transduced. So that's ultimately what this figure is telling you, okay? So it's fatty acids as they are absorbed in the small intestine, the brush border membrane, enterocytes, because of all of these receptors associated ultimately in vagal afferents, leading to the signaling for CCK and GLP-1, leading to appetite control, and also that's directly in the brain, right? Because of what it's doing in the NMDA receptor I just mentioned, and also ultimately the gastric emptying. Okay. Now, that's not enough. CCK regulates gallbladder contraction and secretions from the stomach and the pancreas. Secretin buffers the acidic chyme in the intestinal lumen by stimulating water and bicarbonate secretion, and that enhances even more CCK effects on the pancreas and gallbladder. So you need to have this bicarbonate and this water being secreted. Okay, you need to have the gallbladder now at play. Okay, and when you get that, the CCK can be effective because it's a control over pH balance, what the bicarbonate's doing. It's also allowing for absorption because of the amphipathic nature of what happens when you make bicarbonate. All right, that's increasing the amount of CO2 in the blood, and that all is going to regulate through and move through the GPCR. Now, a number of cellular surface receptor fatty acids are talked about, including the G protein coupled receptors. And the scavenger receptor. It's called scavenger because at the beginning we didn't know what it was. So we called it scavenger, which was another name for orphan receptors. And we know now it's CD36. They've all been identified. And we know that long-chain fatty acids are recognized by GPCR 120, GPCR 40, and CD36. All three of those receptor proteins are receiving fatty acid messaging, as we just saw on the previous slide. Now, let's go in. Let's now pull the lens close and look at CD36. CD36 facilitates cellular fatty acid uptake and it mediates fatty acid-induced signal transduction. We just showed you that. And that does so by influencing cellular calcium for release of neurotransmitters and both arachidonic acid and prostaglandins. Now, this is way more than you need to know probably, but I want you to understand that this whole system, okay, leads to multiple control of neurotransmitters in the gut and the brain. Okay, the gut and the brain. Now think about what that might mean when you alter neurotransmitter signaling when drugs are taking, let's say, under the pharmacological administration of psychiatry or psychology. Okay, think about what that's going to do to this nutrient-stimulated associated appetite control. You think about some psychotropic drugs in causing increases in weight gain. It's because they are antagonizing or maybe agonizing the system. See how the gut can then be regulated by taking psychotropic drugs. Even in taste cells, fatty acid sensing is by CD36 mediates fat perception. So when you taste something, you can taste fatty acids, and preference of foods. And it induces the cephalic, that means the brain, phase of digestion. So when you sense a fat in the diet, and that fatty acid is something that's pleasing in the diet, so it's usually a medium chain or longer chain, then that is also sent to the brain, and that's going to work through the dopaminergic reward pathway and make you feel good that you're consuming that, right? That's why it feels good to eat something like ice cream that has a lot of fatty acid in it, or even potato chips. Okay. All right. In humans, oral fatty acid detection thresholds are higher in subjects carrying single nucleotide polymorphisms that reduce CD36 levels. So your detection threshold, okay, is higher when you have mutations that reduce CD36. So that even has a direct effect then on how fatty acid signaling is transduced when you have a simple mutation in that protein. And there are mutations that have been mapped to CD36. Yeah. So CD36s and the G protein coupled receptors have distinct distribution patterns in the intestine. CD36 is abundant in the proximal small intestine and the villi and enterocytes, that's where absorption occurs, where it contributes to fatty acid and cholesterol uptake from the diet and to signaling events important in initiating chylomicron. Now, chylomicrons are your very first lipoproteins made in the small intestine to package dietary lipids to send them into the liver. Okay, and also into the bloodstream, where that's the initial phase of fat utilization in the blood from chylomicrons, turning into chylomicron remnants, binding back to chylomicron remnant receptors in the liver. That's all part of the initiation of circulatory lipoprotein metabolism, where it starts off in the small intestine. So CD36 plays a role there. Okay. That comes from a paper in FAST Journal 2013, by the way. All right, now I'm just jumping real quickly into a paper. Based on its ability for fatty acid sensing and signaling transduction, CD36 might contribute to the regulation of those enterocyte peptide release. Now, we already have been saying that. However, the possibility has been unexplored until the present work was just done. So using mice that don't have CD36, double negatives, and then the wild type, which have both copies of the gene, the mechanisms underlying CD36 mediated effects and demonstrated involvement of actually cyclic AMP, another component of the system, and calcium were examined. What basically this all found out was CD36 mediates fat perception in taste bud cells and the initiation, again, as we mentioned, the cephalic phase of digestion, influence intestinal fat processing and chylomicron formation, so digestion and packaging into lipoprotein, mediates gut peptide release in response to dietary fatty acid. So I'm not going to go through all of these specific components of this study, okay, because I've already been way too long. But I just want you to know that CD36 and in conjunction with CCK and secretin, those two hormones, they influence satiety and energy homeostasis. We've already talked about that in the general. This paper just tells you a little bit more detail about how it's functioning with cyclic AMP and calcium. If you want to know more about that, you can read this slide. So, the role of this protein, which is also known as FAT or fat, in fatty acid sensing, energy and glucose homeostasis, and regulation to diet-induced obesity and diet-restricted rats. This paper came out, American Journal of Physiology. I told you physiologists were okay by me, in 2015. Now, hypothalamic, that's in the brain, the hypothalamus, fatty acid sensing neurons alter the activity via fatty acid translocator receptor, and that is CD36. It's up there in the hypothalamus. Depletion of the ventromedial hypothalamus, the VMH, of the VMH CD36, so we can specifically target the ventromedial hypothalamic CD36 activity. That leads to a redistribution of adipose stores and insulin resistance. So that means the brain is regulating insulin secretion, right? And also adiposity. We already know that, but now we know it's happening through CD36. Okay. And we know it's happening in the ventromedial hypothalamus. So this whole study assessed the requirement of the VMH, right? That is the ventromedial hypothalamus, CD36 mediated fatty acid sensing coming in from, either the vagus or from the blood for the regulation of energy and glucose homeostasis in diet-induced obese and diet-resistant rats. Now, there was a difference between the diet-induced obese and the diet-restricted rats in relation to CD36. They were doing this diet-induced obesity and diet-restrictive to ask whether or not if you induce obesity, does that have a role in how CD36 works? And indeed it did. So these and other results demonstrate that the ventromedial hypothalamus, CD36, localized CD36, mediated fatty acid sensing and a critical factor in the regulation of energy and glucose homeostasis at that position. So I didn't give you the detail of this paper. I just want you to know that that's what we know. CD36 signaling major player in the brain, and we already remember CD36 major player in the intestine for absorption. So we got a gut-brain axis where CD36 is playing two different roles. And we know what's linking them are things like CD36 and fatty acid. Yeah. So, general conclusions on the diabetic pathophysiology. Diabetes develops from a cooperative failure from glucose homeostasis from both pancreatic beta islet cells and brain-centered, what we have been talking about, glucoregulatory system. That's the whole insulin secretion pathway. Through those hormones, we talked about those three, and then the bigger five, all five of them from the small intestine, all right, and including things from the gut too. The GI tract is also recognized as a major player in the revised model of type 2 diabetes pathogenesis, and that's again what we've been talking about. Indeed, the gut represents the initial target of ingestion, mediating the influx of glucose, right? And the other metabolic products such as what? Fatty acids through CD36. The gut produces the initial meal-induced feedback signal to the rest of the body via neuronal, going to the brain, right? And hormonal signals, that means in the blood. So you got paracrine and endocrine effects here, right? The gut-derived peptides like CCK and GLP-1, the two that we kind of highlighted, exhibit both insulin-dependent and insulin-independent glucoregulatory mechanisms. When they're independent, that means it's working in the brain. So the brain doesn't work through insulin dependence. It works through other pathways. The insulin dependence is mostly in skeletal muscle, cardiac muscle, adipose tissue. Remember that for insulin. Okay. All right. That indicates a link between the gut and the brain and even the pancreas, as we've been talking about, the release of insulin from the pancreas. Of course, that's where it comes from. Pancreas is the only organ that makes insulin, unless you have something like a granuloma, where you have a cell mass that becomes a producer of insulin. And that's something I talked about in another Vera Med format. All right. Dysfunction of the gut-brain axis in mediating glucose homeostasis can instantiate, that means start the cause, be the primary cause of, instantiate, and contribute, that means increasing the activity of, to the initial dysregulation of the brain and the pancreatic beta islets, right? Which is all associated with diabetes or, lack of glucose homeostasis. Lipids play a dominant role in glucose homeostasis just via this discussion. Forget about the fact that glucose is converted to fatty acid, right, upon overeating. And it's working through CD36 through that fatty acid uptake through CD36. Dietary fatty acids that are required to regulate hormonal and metabolic homeostasis in diabetes management. So the point I'm trying to make here is that fatty acids in the diet, working through CD36 in the small intestine, are regulating the expression and secretion of things like CCK and GLP. And those then work through the vagus nerve and through the bloodstream to control appetite control in the brain. Also, fatty acids that make it into the brain, and when CCK is functioning in the brain, control things like via neuronal release of neurotransmitters, things like satiety and feeling good, feeling full, feeling relaxed, feeling you don't want to eat anymore, right? Feeling satisfied after a meal. So these are linked to the gut, but fatty acids here, I wanted to emphasize today, are key sensing molecules in the gut because they're the ones that start the whole process of releasing these hormones, regulating pancreatic, islet cell, beta cell secretion of insulin, control over the cell death of those beta cells, and then also in the brain, how they're controlling overall satiety and well-being, right, through the dopaminergic reward pathway, which we didn't talk about much. So all of that then hopefully gives you a lot of time. If you go back and watch this again, or you've already absorbed it all, right? Like your small intestine absorbed all that fatty acid from eating that ice cream you just consumed while you were watching this. Subjective intellection, okay? Now this is melancholia, and this is actually a woodcut from Albrecht Dürer. And you can see his AD down there. So if you looked for anything that Albrecht Dürer did, you can always find his initials in there. And there's the AD. That was the artist here. This is a woodcut. Okay, so he chiseled this out of wood. I, as some of you may know, like to work with wood. In fact, I'm a tree feller. I cut down trees with my chainsaw and split up the wood and burn it in my house to heat my house, right? So I'm not out there deforesting or anything. I'm dropping dead standing trees, but I'm really into that. So I like working with wood too, but boy, I'm no artist like Albrecht Dürer was. Anyways, he made this in 1514, okay? A couple years before Leonardo da Vinci died, right? So anyways, he was always thinking when he did his art. And here he's got melancholia, right? One of the four basic muses, right? And she's sitting there and she's got all the apparatus of Euclidean geometry on her lap. And she's showing her being a very sophisticated, analytical thinker. But around her is all this chaos, all of this stuff laying around on the floor. And there's an infant that's supposed to be indicating like the purity and innocence of not having intellection yet. And how the infant is totally unaffected by the deep absorption of the subjective intellection, right, of our friend melancholia, right? So melancholia is like the doctor philosopher, right, like the PhD, coming to the grips with the fact that we know a lot and we can describe it and we can manufacture an understanding of it, using things like geometrical matrices, right? Back then, that was the science of the time, right? That was the deterministic science of the time, pre-Isaac Newton, right? So Mathematica was considered to be where you'd really get definite answers. And even old Melancholia is realizing that everything is still sort of chaotic because she's not able to take her nice hardcore reasoning and even in her empirical activity and come up with a clear divine wisdom, which is indicated here. I just love Albrecht Dürer. All right. This was a picture of my younger daughter, Charlotte, when she was a little girl, and in her outfit when she was probably, I think, in maybe first grade when we used to live in Colorado. And probably she wasn't doing this then, but I've got her pointing at Vera of Med. So, okay, Charlotte, thanks for doing that today. Charlotte's now a lawyer, actually, in the state of Washington. Anyways, there's one of my email addresses, info@veravmed.com, website, www.veravmed. I'm Dr. Daniel John Guerra, and there's my personal email if you want to get in touch with me to talk about this particular discussion or any other one that I've given. Please contact us if you want to know more about what we're doing, and I'd be glad to do that. Thanks a lot, and hopefully this YouTube will be something stimulating and exciting and fun for you to watch. I guess there's nothing else to say, but Goodbye for now.